In this video, I'm going to do a walkthrough of the 5 different organisms that you're going to look at for Lab 25, which are pathogens that affect the nervous system. This first image that you're looking at here is of the Culex species of mosquito. You'll notice this one looks different from the Aedes and the Anopheles mosquitoes that we observed in Lab 24. Culex is typically brown. They are smaller than the Aedes and Anopheles mosquitoes. Their antennae are not typically as long. And Culex mosquitoes spread a variety of different diseases, but the most prominent and well known and probably well studied is West Nile virus. When West Nile virus kind of came onto the scene probably almost 20 years ago now, this was really the first time that we studied Culex in detail, because typically the mosquito-borne diseases that we had tracked in the past, like malaria or yellow fever, were transmitted by the Aedes or the Anopheles mosquitoes. So this was a new kind of a new study for us. So this is Culex, again, a little bit smaller, brown in color compared to the Aedes and Anopheles mosquitoes. The next one that we have here that we're looking at is Naegleria fowleri, which causes a disease called PAM, or primary amoebic meningoencephalitis. It's almost always fatal. Fortunately, it's pretty rare. This particular pathogen is a protozoan, an amoeba, and it lives in typically fresh water. And what happens is if you inhale water, then these pathogens, these parasites, can make their way to the brain. So what we're seeing here is cerebrospinal fluid that has been stained, and this is the parasite right here. This is the Naegleria fowleri parasite. You can see this really dark staining structure in the middle here. That's going to be its nucleus. Sometimes you'll see flagella. There is a flagellated stage of Naegleria, so sometimes you might be able to see flagella associated with it as well. So where we typically are going to see Naegleria, we'll see it in the cerebrospinal fluid. We may also see it in brain tissue. And so you'll notice that the structure is going to be different than cells. You know, cells are going to be circular, and this protozoan here is going to typically be oblong and oval in shape. So that's Naegleria fowleri. The next one we're going to look at here, this is a blood smear. So all of these circular structures you see here are blood cells. And then you see these little wiggly structures here. This is Trypanosoma brucei, the causative agent of African sleeping sickness. There is a similar species of trypanosoma called Trypanosoma cruzi, which causes Chagas disease, and they look almost identical to one another. But what you'll see here are these little structures here. They have this little flagella here, this what looks like a little eye spot there on their head region. So this is Trypanosoma brucei, and this is a stained blood smear. That's why they appear purple. But if someone is suspected of having African sleeping sickness or Chagas disease, they would take a blood sample, take a little bit of that, smear it on a slide, stain it, and look for what these are called mastigotes or epimastigotes of Trypanosoma. Now, this particular species of Trypanosoma, Trypanosoma brucei, is spread by the tsetse fly. And so this is a very enlarged image of the tsetse fly, where you can see its head region here. You can see its proboscis extending out here. The tsetse fly is a fly. It's a dipteran, just like, you know, house flies or horse flies, but it is the biological vector for the Trypanosoma brucei. You know, when we talk about vectors, and we have, you know, mechanical vectors and biological vectors, normally flies like horse flies and house flies are mechanical vectors. They land on something that's contaminated. They get the bacteria or the pathogen, whatever it may be, on their wings or their feet, and then they land on something else and they're transferring it that way. But with biological vectors, you know, it becomes a part of the vector. And so the Trypanosoma is like this. The tsetse fly serves as a biological vector for African sleeping sickness. And the last one that we have here in lab 25 is the Taenia solium cysticercus. Now, we looked at Taenia pisiformis in lab 22, and we were able to see the structure of the worm and the different parts of the worm. So what we're seeing here is a particular part of the life cycle called a cysticercus that can become embedded in the muscle of the host, like pork or beef. And what you're seeing here is kind of this curled up structure. Now, this is going to be different than a regular tapeworm disease, because what happens is this can migrate to the brain and cause a disease called neurocysticercosis. And we primarily see this with Taenia solium rather than Taenia pisiformis or Taenia saginata. But what you're seeing here is essentially this form of the worm, and this arrow here is pointing to this, what would become the scolex if this was to hatch out of this form. So that's lab 25, diseases of the nervous system.